Welcome to AP Biology. Today I want to talk about Mendel's law of independent assortment. We're going to be looking at dihybrid crosses for this. So for a dihybrid, we're looking at two traits. So in this example, we're looking at eye color and hair color. And so this is going to be simplified because eye color and hair color both are probably polygenic, but that's okay. We're just using this as an example. So let's say that little b gives you brown eyes, and big B, and let's say little b gives you blue eyes, even though it's more complex than that. And let's say for hair, let's say big H gives you black hair, and little h, let's say, gives you red hair. Okay, so these are the traits. And then if you look at this individual, you can see they're diploid because there are homologous pairs. And this person has a big B and a little b on these homologous chromosomes, and maybe a big H and a little h on these homologous chromosomes. So the genotype of this person is big B, big H, little h. So they would have brown eyes and black hair. Okay, so when they make their gametes, the chromosomes can line up this way, so that maybe this person had originally gotten the black hair and the brown eyes from, let's say, mom, and let's say dad gave this person blue eyes and red hair alleles. So when they separate out, this gamete is going to have the big B and the big H, which code for brown eyes and black hair. So assuming that there's only two alleles for each one, no matter what other alleles they get from the other gamete at fertilization, this baby will have brown eyes and black hair. And this one actually is getting the little b, right, and the little h. So that's for blue eyes and red hair. And so now it depends on what the other parent gives them. But if the other parent gives them also the recessive, then this baby could have blue eyes and red hair. They don't have to line up like this, though. Mendel's law of independent assortment says when the genes that we're looking at are on different chromosomes, they can line up however the heck they want. So if this is a big B and this is a little b, the big H doesn't have to line up on the same side as the B. The big H could line up over on this side, and therefore the little h would be here. So the homologous chromosomes can sort of go in whichever order they randomly end up. And so you could get the big B and the little h. So that's a different combination than what we saw over here, right? This is brown eyes and red hair. And this one would be, let's see, the little b and the big H. So this is also a different combination. It's the blue eyes and the black hair. And so it would depend on what the other parent was donating. But you can see these gametes are all different. They're all donating something different to the baby if they were to get fertilized. So alleles for different traits are distributed to sex cells and then to offspring independently of one another, but that's only if they're on different chromosomes. So the terms we're going to use, we're going to say they sort independently, or we get independent assortment, but only if the genes are on different chromosomes. So if they sort independently or have independent assortment, if the genes are on different chromosomes, we are going to say they are unlinked. And so that's what this whole section of genetics is about. If the genes that we're looking at, hair color and eye color, are on different chromosomes, then they can be distributed like however, right? You can get a big B, if you had this person, big B, little b, big H, little h. The big B could end up with the big H, right? So the gamete here is only going to get one of the B's and one of the H's, but which one they get sort of is just random, right? The baby could get a big B and a little h. Or the baby could get a little b with a big H, or they could get a little b with a little h. Any of those combinations could happen. So let's say in a dihybrid cross with two traits, let's say we're looking at the shape of the pea and the color of the pea. So here are a bunch of peas. They're all green and they're all round. So let's say big R is for round and little r would be wrinkled. And this picture doesn't actually match my, my genotype here because, believe it or not, big Y is yellow. So yellow is actually dominant in peas. And then little y would be for green. Okay, so my picture doesn't match, but that's okay. So what would happen if this, if a plant with this genotype mated with a plant with this genotype? It could be self-fertilization or it could be a different plant. But in either case, how do you figure out the gametes? So you could your R's have to segregate, right? So you could get a big R, but you wouldn't get a big R and a little r. That would be like giving the baby both of your homologous chromosomes, right? You've got a big R and a little r. The baby's not going to get both. They separate. So the baby can only get one of the R's and one of the Y's. So the baby could get a big R and a big Y, or it could be a big R and a little y. 50-50, right? If you get a big R, it's a 50% chance as to whether you get the big Y with it or the little y. Or the baby could get the little R with the big Y or the little r with the little y. And to help you make sure that you don't miss any of these, the way to do it sort of systematically is to FOIL it like you do in algebra class. So the first is big R, big Y. O is for outer, big R, little y. And then I is for inner, little r, big Y. And then little r, little y. Be careful that in each gamete you only have one of each allele. When people start doing these, sometimes they mess up and they'll say one of the gametes has big R, little r, and one of the gametes has big Y, little y. But that's absolutely wrong because your baby, this baby, would be getting two sets of two alleles for the color, but nothing for the shape. And that's not right. You got to separate those out. Okay, so what would gametes for each of these make? So let's say, what gametes would each of these make? So let's say this parent and this parent got together. What gametes would this one make? Well, the first would be big A, big D. Outer would be big A, little d. Inner is big A, big D. And then last is big A, little d. Notice that this is a double of that and this is a double of that. So I can make a huge Punnett square if I feel like it, but I really don't have to, which is kind of nice. So, and then this one, I'd get a big A and a little d, and a big A and a little d again, and then a little a and a little d. I can FOIL the whole thing out if I want. So this one's gametes would be, first would be big A, little d. For, let's see, we FOIL it, right? Outer would be big A, little d. Inner would be little a, little d. And then last would be little a, little d. Notice the repeats. If you don't want to make that big of a Punnett square, you don't need to. So if you have two of these and two of these, it's okay to just write one. So although this is a dihybrid cross, it's not one of those, I don't have to do one of those huge Punnett squares like I would have to with the last example. So these babies would look like this. I messed that up. Oops. All right, big A, little d, big A, little d, little d. Are these right? Big A, big D, little d. Yep. All right, so they would all have at least one big A, and these would have big Ds, and these would look different. So these would have the same phenotype as long as it's just complete dominance, and these two would have the same phenotype. So the genotypic ratio would be 1 to 1 to 1 to 1 because they're all different. But the phenotypic ratio would be 1 to 1, right? Or 2 to 2 if you want. Okay, so let's do some examples of this. In pea plants, yellow color is dominant to green, so I'm going to write my knowns. I'm going to say big Y is yellow, and little y I'm going to try to write differently. That one's going to be green. And round shape, big R. Round shape is dominant to wrinkled, so little r is going to be wrinkled. And so the reason that you'll see crosses between pea plants is that this is what Mendel worked with, so a lot of times we still give a lot of practice on this. A dihybrid cross between a true breeding plant with yellow round seeds, big Y, big Y, big R, and a true breeding plant with green wrinkled seeds, little y, little r, results in an F1 generation of plants that are yellow and round. So you get big Y, big R, little r, because this one can only give a big Y, only a big R. This one can only give the recessives. Okay, so now it says, crossing the two F1 plants produces an F2 generation. Show the Punnett square that produces the F2 generation and list the phenotypic ratio. So I'm going to take this one and mate it with itself. So I don't have enough space, so I'm going to actually erase everything and try it again. Let's do green, right? So I've got big Y, big R, little r, and I'm mating it with itself. So we're going to FOIL this. Big Y, big R. When we make the gametes, notice the gamete has one of each allele. Big Y, little r, big R, and little y, little r. So hard for me to just write this with a straight line. All right, be more careful than I am and make sure that your lines are straight and that you truly making your big I are making your big Y's look different from your little Y's is probably a good idea. Okay, so this one is big Y, big R, big R. In fact, all of this row is going to get a big Y and a big R, big Y, big R from this parent, right? From this gamete, big Y, big R. I almost don't even care what they're getting from the other guy, but I don't know. I'll finish it up. Why not? Little Y, big R, little y, little r. Okay, and then this one is giving a big Y to everybody. And then big Y from that one, big Y from that one, and then little y and little y. Okay, little r from the parent on the top. That's supposed to be little r's. Big R from this guy. Okay, bum, 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 bum. I'm going to do all my Y's first. I don't know why. The R from the top. R's from the sides now. It's supposed to be an R. Let's see. Little r's from the side over here, big R from the side here, and little r's here, here. Okay. Super duper. What you should memorize when you do a heterozygous mated with a heterozygous is that you're going to get a 9 to 3 to 3 to 1 ratio. And this is what it's going to look like. Whoa, that was a lot less work. So what you see if we're looking at anybody who has at least one big R and one big Y. 1, 2, 3, 4. This one also 5. This one, this one, this one, this one. 4, 5, 6, 7, 8, 9. I get 9 that are dominant for both the Y and the R. And then dominant for just the R's, I've got 1, 2, 3. And dominant for just the Y's, that's, let's do a different thing. Let's do a circle. Big Y, but little r's for these. So that's 3 of those. And then 1 of the totally recessive. So if you do heterozygous by heterozygous, right, this one mated with itself or with another one just like it, you are going to get a 9 to 3 to 3 to 1 ratio if the two genes sort independently. That means if they're on different chromosomes, if they're not linked.